Our next speaker is Greg Fraley, speaking about the neuroendocrine relationships with affective states. And just as a reminder for our virtual attendees, if you could please type your questions into the Q&A box. Well, we might have some time during the talks, but we also have some time after lunch for a roundtable with all speakers. So with that, Dr. Fraley.
The last few years, my lab is trying to better understand the neuroendocrine bases of positive and negative affective states. And we've been doing this quite a bit. And essentially, for all the cool people in the audience who really know this movie, the Scarecrow here is having a pretty bad day. And the Crow is actually having a good day. Trying to understand what the differences in their brains are, okay? What's actually going on inside the mind, inside the brain. And so I'm going to talk a little bit this morning about the stress response that Dr. Kensel has already given an excellent overview of, so I won't spend much time on that. Oh, I got rid of that. I was going to talk about the fear freeze and neurobiology underlying the fear freeze, but I realized I misinterpreted Dr. Weimer's instructions and don't have enough time, so I'm not going to talk about that. But I want to talk about two important neurotransmitters called dopamine and serotonin and what their involvement is on associated with aggressive behaviors or emotional mood states. And I'll go over some research that we've been doing in the lab. I focus on ducks because that's just what I like, but I have similar experiments going on with broilers, layers, and turkeys. And the pathways, the neural pathways I'm going to show you are actually very highly conserved. And a lot of this work that I'm going to talk about actually started back in the 1970s and 80s in mammals. And so we're just kind of catching up with that in birds. Okay. And I like to show you lots of pretty pictures of birds because I like birds. All right, so Dr. Kensel's already given a nice overview of this. And so when an animal undergoes some stress, there are actually two different components to that stress response. One is the sympathetic system, which involves mostly epinephrine and norepinephrine. And the other is this septal-hypothalamic pituitary adrenal axis that Dr. Kensel's already given us a great overview of. And, okay, so I changed this last night. I must not have saved it. I'm going to skip this. Sorry. This is very, very cool. This fear freeze, a lot of people refer to it as a part of the sympathetic nervous system. The sympathetic nervous system is a component, but the fear freeze itself is a very complicated cognitive neural pathway. And when an animal is presented with a stressor, there are different possible behavioral outcomes, such as going into fight or flight, which is sympathetic, or the fear freeze, which is purely cognitive, purely brain pathways, or going into tonic immobility. And the neural pathways between freezing and tonic immobility are controversial about whether or not it's similar and what the relationship is between them. But that's something my lab is starting to also look into. And there's an absolutely fantastic review about the neurobiology of the fear freeze out of Nature Reviews Neuroscience. And this is figures from this. So when I'm, as a neuroscientist, when I look at this, I'm like, oh yeah, I get this. This makes sense. However, these figures from this review make my brain hurt too. Okay, but essentially what this is telling us is during the actual fear freeze, which is on that side, your left, these blue pathways are dominant, and this is what's causing the animal to freeze. And this animal, at that same time, the parasympathetic nervous system is dominant. Okay, because a part of this fear freeze is to increase awareness of its environment, which causes pupil dilation. Okay, so that is parasympathetic. It also keeps the animal still and quiet. So the parasympathetic nervous system during the fear freeze actually stimulates a reduction in heart rate and a reduction in respiratory rate. Okay, because the idea here is we're trying to freeze to stay away from the predator that's confronting us. And you can't do that if you're going, right? So the parasympathetic system actually acts to help calm the animal as well as increase conscious awareness of the surroundings. And then if a additional stimulus from that predator, like moving towards a prey, then the sympathetic nervous system becomes dominant, and that's over on this side. And then that allows the fight or flight or whatever, usually flight. Okay. So the sympathetic nervous system and parasympathetic nervous systems are both very important components of that fear freeze, but during the actual freeze, the parasympathetic is actually dominant. Okay. But I'm not really going to talk about that anymore. So I wasn't going to talk about it at all, but it just came up. So I want to talk about affective states, and I am going to focus on ducks, but again, these pathways are very highly conserved. And if you see, and I'm eventually going to be talking about these open water systems that are given to ducks in commercial barns. And one of the things that we're seeing is thought that this creates a positive affective state, and that this is allowing them to perform natural grooming behaviors, preening behaviors, head dunking and such. But what we're also finding is under certain types of management, mostly academic management systems, we're eliciting a negative affective state. Okay. I hate PCs. I really hate PCs. Yeah. So we're actually creating a system where a subpopulation of the ducks in the pens or in the barns start resource guarding and become real jerks. All right, so how do we measure this affective state? So one way is through behavior, and that is an absolutely excellent way. However, as a neuroscientist, I think behavior is the beginning of knowledge. It may not necessarily be the end. We can also do cognitive tests, cognitive bias testing, which is extremely cool. And I'm starting to work with folks like Dr. Weimer so I can learn more about it, because right now I don't know anything about it. But we can also assess the neuroendocrine states. And like I said, this has been a field that's gone back to the 1980s, looking at changes in dopamine and serotonin activity in relationship to different emotional states of animals. And again, this is fairly well known in mammals, not so well in birds, but the pathways are fairly highly conserved. So with serotonin is generally believed to be involved with mood regulation, some aggression, but sleep, memory, lots of different things, including social behaviors. And low levels of serotonin activity in mammals is related to a depressive mood. Okay, and this is something all of us, especially in the United States, are very much aware of. The serotonin reuptake inhibitors, so the drugs that, like Prozac, that increase serotonin activity, are associated with increasing affective state in people. Okay, so serotonin is very strongly implicated. An increase in serotonin activity is associated with a positive affective states, and this has been shown in many different mammals as well. Dopamine is also involved with emotions, reward, motivation, and also very important with aggression, as well as learning and movement and even reproduction. So inconsistent or increased dopamine activity in certain areas of the brain is very closely related to aggressive behaviors. You can give pharmacological manipulations to highly aggressive chickens and either remove that aggression by inhibiting dopamine or stimulate aggressive behavior in genetically selected non-aggressive birds, chickens, by giving dopamine agonists. Okay, so it's very well established relationship between dopamine and aggression. So I want to talk a little bit about the biosynthesis of these two neurotransmitters, because it's going to be really important for the rest of my talk. So dopamine and serotonin are both synthesized from either tyrosine or tryptophan, respectively, the amino acids. And they are synthesized into the active hormones through slightly more complicated pathway, and I'm showing you, but essentially by tyrosine hydroxylase and tryptophan hydroxylase for dopamine and serotonin, respectively. It is very important to understand that the expression of these enzymes is the rate-limiting factor for producing the neurotransmitters, dopamine, okay? You can give bucket loads of tryptophan or tyrosine in a diet, and it won't change how much serotonin there is in the brain unless you increase the gene expression of these enzymes, synthetic enzymes. It is also important to understand that for tryptophan hydroxylase, that enzyme is produced by two different genes, TPH1 and TPH2. It was originally thought in mammals that TPH1 was important in the brain, but it turns out it's not. It's important in the pineal gland and in peripheral tissues, production of serotonin. And in the brain, TPH2 in mammals is predominant for producing serotonin in the brain. These neurotransmitters become activated, and then they can be broken down into respective metabolites. And when we're looking at activity, we ultimately are going to look at the ratio of metabolites to neurotransmitters, okay? And it's what we call either the dopamine or serotonin turnover. It's not a rate. This is not per unit time, but just the turnover. It's a ratio of metabolites to neurotransmitters, okay? All right, that's just to remind me to talk about this carefully. So this synapse, okay, so up on top is the presynaptic membrane that's going to make, just using serotonin as an example, tryptophan is going to be packaged by the smooth ER with all of the synthetic enzymes into a vesicle. That's transported down the axon where the synthesis actually occurs. It's stored very, very shortly. And upon release, upon release into the synapse, several things can happen. One, it can bind to its receptor on the postsynaptic membrane and activate whatever cascade is going to go on down the road. It can be reuptaked or recycled into that presynaptic membrane by what's called a selective serotonin reuptake transporter, okay, which is what SSRIs like Prozac inhibit. And once it's been recycled, it can either be repackaged and re-released into the synapse, or it can be broken down by enzymes like monoamine oxidase. That is unfortunately not the complete story. There are also serotonin receptors on the presynaptic membrane where serotonin can bind to them and completely impact the expression of the synthetic enzymes, of the reuptake mechanisms, of the release of serotonin. There are also these glial cells. Okay, glial are the glue cells, the support cells in the nervous system, and they surround this synapse, and they too participate in the clearance and removal of these neurotransmitters. Okay, so really, really complicated. So this is one of the reasons why we tend to not look just at absolute levels of dopamine and serotonin, because that can be deceiving because we don't know where that dopamine is, but we're looking at the ratio of metabolites to the neurotransmitter itself to try to understand what the activity rate is of that neurotransmitter, okay? And hopefully that'll make a little bit more sense in a minute. But these neurotransmitters, dopamine, serotonin especially is made pretty much in the midbrain, okay? In these two areas, the caudal and rostral midbrain. And serotonin sends its axons absolutely everywhere in the brain. Serotonin has its little serotonergic fingers in just about every physiological homeostatic process in the brain, okay? And dopamine's the same. Dopamine is also made in the midbrain. Unfortunately for us neuroscientists, it's also made in the hypothalamus and other places, and so that gets a little wonky for us with interpretation, okay? Now this turnover, again, this is very, very important. What we're looking at is if we have an increase in serotonin activity, that means that it's going to be a low turnover. Okay, so we have lots of serotonin, very little bits of lower levels of metabolite. If we have a high turnover, that means we have relatively low levels of serotonin, but high levels of the metabolite 5-HEA. Okay, so these turnovers are kind of counterintuitive. So a low turnover means high activity, synaptic activity, or suggests high synaptic activity, whereas high turnover refers to low synaptic activity. So if we increase dopamine activity, for example, that has been well established in mammals and birds to increase aggressive behaviors. And again, these are the equations down here at the bottom that show how we calculate all of this. Okay. And yeah, I just had to point out, this is a northern oriole bathing in water. That'll make sense later. We also use QRT PCR to look at gene expression of these synthetic enzymes. Now, I told you that TPH1 is not in the brain in mammals. It's mostly TPH2. However, working in the duck, we don't know that for sure. So whenever we do QRT, we're actually going to measure both of them. Okay? And we also look at CRH, as Dr. Kenzel has already well described as a part of stress components and welfare. There's another neuromodulator in the brain, the hypothalamus, called neuropeptide Y, that's typically associated with feeding, but is also known to innervate and regulate CRH neurons. We look at it. And of course, Dr. Kenzel talked about AVT. And in the duck, we're not able to look at it. I know it's there, but the genes for the duck, the gene sequences for the duck kind of suck, the genome. Actually, I take that back. The chicken genome sucks. The duck genome really, really sucks. And so we have been trying for a year all kinds of different sets of primers to look at AVT expression in the duck, but we have not been able to validate a single set yet. But we'll keep trying. All right, so the first one, we want to look at these things called preening cups and to see if they are really increasing or improving the affective state of these birds. So Peking ducks are, of course, waterfowl. They are domesticated mallards. And there's literally a global controversy as to whether in a commercial barn, these birds need water to swim in, bathe in, dunk their heads in, preen in, you name it. And there's all types there's pools, baths, water troughs, misters, preening cups, and these things called Pekinas that I'm not going to talk about. Horrible name. But in the European Union, some type of open water system is required. It's recommended in the US by certain welfare certifications agencies like the American Humane. And there are a lot of positive perceptions about these presence of these water troughs as to whether or not they are allowed the expression of natural behaviors such as so-called wet preening, head dunking, swimming, etc. And there are studies that have shown that they improve eye and nostril health and increase production. There are also a lot of negative perceptions or data where it's not so good, mostly due to bacterial buildup, but it's really poor, produces really poor environmental conditions. And some studies have shown an increase in mortality and decreased production. And the differences between the decrease or increase in production is, I'm sure, purely about experimental design. So these preening cups is what I'm going to focus on today, and these are essentially giant upside-down mushrooms that contain water. The water is constantly flowing, and usually when the ducks bump up against it, they'll dump it upside down to help clean it out. And in a commercial setting where these are required, it's usually one of these per 1,500 to 2,000 ducks. Okay. In our study, we were using an academic setting, and we wanted to really, really ramp it up, right? Because one of the things that we neuroscientists do is we want to see how some environmental change or something affects the brain chemistry of an animal. We absolutely whomp them over the head with it. So we really, really are pushing the concentration of preening cups per ducks to exacerbate the situation. whether positive or negative. Okay. And essentially, we had 2 rooms, 2 pens each room, and one room received the preening cups, the other room did not. And we did that intentionally. We didn't have one room with and without for the 2 rooms, because one of the things, other studies going on in the lab is about vocalizations, and we know that ducks, while they are using these preening cups, elicit very specific vocalizations. We don't know what they mean yet, but we're working on it. But we were very concerned about if we had a pen with and without in one room, those vocalizations would affect the behavior and affective state of the other birds. So we separated the rooms, but then we repeated it 3 times, swapping the rooms back and forth, okay, to deal with the confound. Preening cups at the time were placed on day 18 of production and grow out meat peking ducks who have a production period of about 42 days, 35 to 42 days. And on day 18, we collected a set of ducks and assayed lots of things about them, including collecting brains. And then immediately after that, the preening cups were placed into the room. And we repeated the collection and brain collections at day 43, okay, which is the end of the production period for that flock. So we actually have 2 sets of controls. We have a cross-sectional set of ducks with and without the preening cups in different rooms. We also have a longitudinal control, so we're looking at the ducks before and after the preening cup placement. Okay. Now, in order to do the brain analyses, we collect the brains, they're immediately frozen. We have to microdissect them. You can't take a whole brain and grind it up and measure dopamine, serotonin, because you're going to get so much dilution of effects, because again, every part of the brain has a different function. And so this is what I call my redneck CT scan, because it's not a CT scan at all. But again, we're looking at that caudal mesencephalon, rostral mesencephalon, diencephalon, and the forebrain, the telencephalon, what I like to call the thinky parts. Okay? And we microdissect them and process each of them as a separate unit. And again, we're looking at both. Oh, we do that on one side of the brain. On the other side of the brain, we do similar microdissections and process them for QRT, for the synthetic enzymes. And we do a combination of HPLC and mass spec for the neurotransmitters and all of their metabolites, and QRT PCR for the mRNA expression. All right, so here we're going to look at serotonin. Absolute levels are on your left and turnover on the right. And we don't really see much happening at all when we look at the pre-placement condition versus controls and preening cup placed ducks. Don't really see much. So based upon this alone, remember, an increase in serotonin turnover means low activity, which we know in mammals and birds, that's a negative affective state. So right now we have no evidence of the preening cups impacting a positive affective state. When we look at dopamine, we do see a decrease in dopamine turnover associated with the preening cups, okay, which suggests an increase in dopamine activity, which can be related to aggressiveness. And as you saw from that video, these ducks were very aggressive. And we had all kinds of physical signs of their feather pecking to the point of drawing blood and even occasionally killing a duck, another conspecific. Now, when we made these primers, like I said, the duck genome is difficult to work with. We validate these things to death, including taking the amplicons, isolating the amplicons in a gel and getting them sequenced and then researching the sequence, right? Everything we can to validate these primers. And despite the fact that in mammals, TPH1 is not expressed in the brain, we see it. And that's annoying because that makes now we're seeing both TPH1 and TPH2 being expressed in the brain. And this is in large areas of the brain, so I haven't mapped it out yet, which is another problem. But we are seeing some changes, but it's not really matching up to what we're seeing with the mass spec. And we are convinced now, and I'm going to show you this over and over again, but we are convinced that this is because of timing. So the best time to sample a brain to measure QRT is going to be different than the optimal time to sample a brain for doing mass spec for neurotransmitters. Okay, so that's a little problematic for us. Okay, and it doesn't matter what brain region, we're just not seeing much effects on the synthetic gene expression. Doesn't mean it's not there, we just didn't see it. And this forebrain, this is a very large part of telencephalon. And this is the last time I'm going to show a slide about it because we have now been convinced after a few studies that we need to microdissect that forebrain down into smaller bits, depending upon more functional relationships of the cortex, that telencephalon, with different behaviors. So essentially, we saw these age effects that coincided with the placement of preening cups. We didn't see it with the control ducts, and so we were pretty convinced that that resource guarding that we saw with the preening cups was associated with this increase in dopamine activity. Okay, again, supporting literally decades of research. Okay. So, but that again, this is in our little academic setting. So how does that translate to a commercial barn? So the next step, this is what I just said. Our next step is we went to the commercial farm and we tried to replicate this as best we could. Again, this is now in groups of barns of 5,000 to 8,000 birds. Okay, and they're placing about one preening cup for every 1,500 birds. And at this time, this is 2 years after we did the first study, and this study has actually just been accepted with revisions. And Dr. Toscano, if you're listening, that was submitted a couple of weeks ago. And they moved the placement date up from 18 to 22 days of age. And this is an AHA certified humane company, stakeholder. And to maintain that, they can't have barns without the preening cups. So we have that, and we can't go to another stakeholder because we're going to have differences in barn design, management styles, genetics, too many confounding issues. So we tried to do this as a longitudinal study where we, again, collected birds and sampled birds just the day before the preening cups were placed, and then at 2 different ages afterwards, leading up to the end of the flock. We also did, we assayed and measured lots and lots of stuff with doing transect for body condition scores, for welfare, litter samples, and blood. I mean, you name it, we collected it. I don't have time today to go over all of it. And again, we did the same brain dissection, but this time I'm not going to focus on forebrain, just the other 3 areas that are more relevant towards affective state at the moment. And again, we're slicing the brains in half, doing mass spec on one side, QRT on the other. And yeah, we did statistics. Lots and lots of statistics. So I don't have time to go over all the transect data that we have, and the current trends are looking at these probabilities of various body condition scores, which I'm sure is the best way, but the data illustration is wretchedly bad. But suffice it to say, all of these birds were just beautiful. Okay, so there were no differences before and after. They were all absolutely pristine, wonderful nostril eye health, clean feathers, and just all beautiful birds. So there were no issues with that. When we're looking at serotonin turnover, again, in the different brain areas, again, Not seeing anything. We're not seeing anything. Oh, I'm sorry. We also sampled birds that were in the barns actively using the preening cups, and those who are on the far side of the barn not using them. Okay? And we're not using that as a control. These neurotransmitter turnover rates are the result of repeated experiences, right? This is a long-term assay. We're not looking at short-term. So just because the bird is using it, we may not, that will likely involve a change in these neurotransmitters, but it's in too short of a time that we can't measure. So we're looking at the cumulative effects. And so because of that, we wanted to make sure we were collecting birds using and not using. And again, with dopamine turnover, we saw this decrease in age. Unfortunately, because we could not sample barns, did not have access to barns without preening cups, we can't say if this is necessarily related to those preening cups, or is it just simply age-related differences? Okay, ducks do become more aggressive as they get older in general, but as I said, these birds had just beautiful body condition scores. We had absolutely no evidence of aggression. And again, the QRT, nothing. It was very difficult to interpret. And so again, we did see a decrease in brain turnover, which suggests an increase in dopamine activity associated with in all of the brain areas, which has been shown to be associated with aggression in chickens and many different mammal species. But we have this problem that we can't tease apart the age versus preening cups. And my students were absolutely wonderful to see that this change in dopamine over age has been reported by me a very long time ago when I was in Wayne's lab, and as well as other students that he had. But a key difference is that when we were doing it, we were looking at brother beaters for one. We were looking at changes in dopamine from hatch to puberty. The changes in dopamine that we're seeing with these ducks, with the preening cups, is only over 2 weeks. And these birds are nowhere near puberty, right? So cannot rule out the simple age-related differences that we're seeing, but I think it's unlikely. But I don't know that yet. All right, so again, once again, the dopamine and serotonin, even though serotonin is so strongly implicated with positive affective states, the way we're measuring, the methodology we're using, we just can't seem to tease that out. Okay. Aggression, we can see, but the positive affective states, not so much. All right, so we're like, okay, well, what if we really do something to promote a negative affective state? Can we then see changes in serotonin and dopamine? So we looked at transportation, which has already been mentioned, is really stressful. Okay, so ducks are considered by the USDA herd animals, so they are literally walked down the length of the barn, walked up a ramp, and then they put themselves on the semis. They are put into small cages or section of the semi so that they are restricted in a crate-like manner, but they walk themselves into it. Okay, and there are all kinds. There's absolutely nothing about this process that we can't just admit is stressful. Okay. So with this study, we're looking at the brain chemistry, and we're trying to associate that or relate it to changes in the septal hypothalamic pituitary adrenal axis that Dr. Kenzel's already talked about. And one of the things, we don't like making these birds unhappy. We don't like intentionally stressing them out. So instead of saying that we're really stressing or trying to stress these birds out, I like to think of it as we're just giving a day at the spa. Okay, that was really funny in my head, but okay. Well, Colin Skanes did a review years ago looking at different effects of transportation and shackling on port and heterophil lymphocyte ratios, where some studies show an increase, some studies show a decrease, other studies show no effects at all. And we had thought that maybe that was really about the difference in and sampling relative to that bird's perceived onset of stress. So my lab has literally spent years reassessing the duck's time course of response to stress. And we've done pharmacological manipulations like Dr. Kenzo talked about with injecting CRH or injecting ACTH, the anterior pituitary hormone, and then measuring the hormones. And we've done it with following management environmental stressors like food deprivation that Wayne has also done. So we have nailed down this time course. Okay, so essentially what we did, we went to a commercial facility, and these were 23-week-old Peking duck breeders. This is a young adult. They're just about to come into peak. And we had controls, which were ducks that we just grabbed right off the floor and euthanized and did our collections. We couldn't do the whole herding and walking onto the crated trucks. So what we did is we took some and we walked them, picked them up, put them in the crate, and those birds just stayed right there in the barn. And then we had some that we did the same thing, crated them up, put them in the back of a pickup, and my graduate students found this drunken truck driver at a bar somewhere who just drove them around on a nice sunny fall day, relatively cool for the Midwest United States. And then immediately the birds were euthanized and samples collected after that 90-minute period. Okay, and that 90-minute period we have shown will give us peak glucocorticoid levels in both drakes and hens. Okay. And one thing that always comes up, especially with reviewers, is, well, how long did it take you? What's the impact of your actual catching and collecting? Let me just say that we're really, really good at this. We can walk into a barn, grab a bird, get that blood sample, get it euthanized. We use pentobarbital to euthanize, and get that brain out literally in less than 3 minutes. Okay, so we have minimized that as much as we possibly can. We send blood smears off. We can measure them, but it takes forever for us. So we send them off to avian pathologists at the Antac Diagnostic Lab who are unaware of the treatment groups and just send back the counts for us. And again, the same areas, same statistics, all similar. So court levels is really, really interesting, and we're seeing a sex difference. But it's contrary to what Dr. Kenzo discussed. We are always seeing that the hens are much more responsive to stressors in terms of glucocorticoid release and glucocorticoid and HLR. Okay, and the hens being more responsive than the drakes is actually agrees with a lot of the mammalian literature, which I'll talk about. But in the hens, okay, the crating alone caused a significant increase in court. Then it was really increased following that transportation, but the drakes did not show that physiological stress until transportation, with either the court levels or HLR. But the hens showed it immediately, or not immediately, but associated just with crating alone. If we look at the serotonin turnover, we're getting the same type of pattern. The hens showed the stepwise increase in serotonin turnover, which means a stepwise decrease in serotonin activity. Okay, and again, that decrease in serotonin activity is associated with decreased welfare or a negative affective state. Okay, and we just didn't see any changes at all in the drakes. With the dopamine turnover, we saw absolutely no differences. And so this suggests that, and the body condition scores and such that we have collected with them and other parameters I don't have time to talk about, they weren't showing any aggressive behaviors. They weren't in those crates pecking at each other, attacking each other, biting each other, plucking feathers from each other. They were just sitting there. Okay, and that lack of aggressive behaviors is reflected here as well. Okay. With the QRT, just absolutely nothing, no differences whatsoever in any brain area, any primers. Okay, so we got this wonderful increase in CORT and HLR with a decreased serotonergic activity in the hens. And so again, and I have to tell you, we've seen this with heat stress, we've seen this with food deprivation, we've seen this with ACTH injections, we've seen this over and over and over and over again, that the hens are more responsive than the drakes. Okay? And if we follow the mammalian literature, there's a lot of evidence for this showing estradiol's interaction with glucocorticoid receptors, and that actually enhance that receptor activity. And so that could be what we're seeing here. However, that gonadal SHPA pathways have been mostly ignored in birds. And so that's something we're starting to look at. And again, we're not getting a nice match like we had hoped for between the mass spec for neurotransmitters, the turnovers with the QRT, and we are absolutely convinced that's just all about the timing of collection. There's going to be an optimal time for QRT and an optimal time for mass spec, and we're just not meshing the two yet. We haven't figured out how to do that. So again, is well established in the mammalian literature. There's a sex difference in the stress response. So when you're trying to assess response using CORT or HLR, you have to consider the sex of the bird. Anytime in neuroendocrinology, neuroscience, when we see a sex difference, that just screams and yells at us that there's probably going to be an age difference too. Okay, and if you look at the wild avian literature, that is shown everywhere, where younger birds tend to be less responsive than older birds. And actually, Dr. Karcher has some wonderful data showing that in pullets as well. And again, agreeing with decades of mammalian literature, dopamine and serotonin are valid indicators of a negative affective state, but getting to that positive affective state, improving it, is still kind of elusive to us. So some of the things that we need to think about as we're moving forward is we need to better understand the positive affective state. And as I understand from colleagues, even behaviorally, that's difficult to assess. Neurobiologically, right now, it is as well. And one of the things that I've been starting to look at after some urging from Colin Skanes and working with a colleague at Purdue, Heather Neve, is looking at endogenous opiates and endogenous cannabinoids. And there is some very fuzzy evidence that these may be, whether looking in the brain or in the blood, that these may be related to positive affective states. So something that could give us a better clue physiologically towards the positive. Okay. Another thing my lab is doing is looking at the epigenetics. One of the things that we know in chickens, ducks, and wild birds is that prolonged periods of stress lead to a deposition of glucocorticoids into the egg. And we know in wild bird literature and in ducks, Peking ducks, that parents who are stressed out for long periods of time, that causes a physiological and behavioral change in their offspring. And this is very well established in wild birds, and we just published that in Pekins. And the offspring tend to be more fearful and have an enhanced stress response, physiologic stress response. And so we are now exploring the epigenetics of glucocorticoids impact in the egg, as well as other potential pathways. All right, and so a couple of things, take homes that I want you to remember. One, I'm freaky over birds, but it's really important. You just can't take a whole brain out of an animal, grind up that whole brain, and assay. Okay? Actually, there are one time you can. But for most of what we are concerned about, especially if we're trying to relate brain chemistry, neurobiology to behavior, you have to focus. You need to be able to accurately microdissect these brains. You also can't take these brain data and analyze them in a vacuum. Okay, right? You've got to be able to relate it to environment and behavior. Okay, that's absolutely critical. Once you know all of that, it's easy to connect the dots. But if you don't, interpretation can be challenging. And one very important thing to remember about neurobiology, there is no structure of the brain, no neurotransmitter in the brain, and no neuromodulator in the brain that only has one job. They all have many, many jobs, many of them in parallel. Feeding and reproductive systems are exact same neurons, exact same neurotransmitters, same physical pathways, but doing different things, okay? Having opposite effects. So very, very careful. Interpretation needs to be very careful. And also remember, dose is everything. I've had people ask me, why don't we just give them more serotonin, just make them have more serotonin? Well, keep in mind, here's something we absolutely know about in humans, okay? Right? So that increasing serotonin activity, thus decreasing serotonin turnover, is a $1 trillion global market to regulate serotonin levels. If your serotonin levels are down here, these are the people that are depressed all the time, have suicidal thoughts, okay? Poor welfare, we can say. Well-being, maybe. You take Prozac, you bring that serotonin up to here, I'm happy, or right, I've gotten rid of the huge mood swings, life is good. You knock it up to here, you have anorexia nervosa. You knock it up to here, you're schizophrenic. So we just can't go willy-nilly saying, let's just increase serotonin levels, okay? We need to better understand what the different levels mean associated with very specific behaviors. All right, and like I said, I'm doing these studies looking at brain turnovers with a lot of folks around the country, including with Karen in Turkeys, which we haven't gotten a final answer yet, but we're close, and just starting with Dr. Weimer, and many others looking at all the poultry species that we have ready access to anyway. I'd like to look at emus just because I'm not kind of hairpin, but they're scary. All right, so I have to thank my lab. They're the ones who actually did all the work that I talked about. The names on the left in black, those are my graduate students, and the ones in red are undergraduates who have been working on all of this. And thank you very much, and happy to take questions if we have time.
All right, we actually are right at our time now, but we do have an hour and a half for a roundtable after lunch. So thank you, Dr. Fraley.